Hey everyone, so today we're going to talk about Chapter 9, Microbial Nutrition and Growth, Part 3, the study of microbial growth. So we've talked already about all the different nutrients that microorganisms need and different ways that we can categorize microorganisms based on how they obtain their carbon and their hydrogen, whether they're chemoheterotrophs, for example, or phototrophs or photoautotrophs, that sort of thing. And we've talked about how they get their different nutrients. This section we're going to learn about how we study that. How do we figure out how much bacteria is present and actually count the numbers of organisms? And that may seem like a difficult task if you've looked at a colony and realized that a single colony can have billions of organisms. We don't actually have to count every single one of them, but we have different ways to count bacteria and to study how they grow. And this is important because it's important to know what stage of growth organisms are, because different antibiotics, different treatments, that sort of thing will work better when the cells are actively growing as compared to when they're in a stationary phase or when they're not growing as efficiently. So let's get on with this talk. So these are the learning outcomes. Summarize the steps of cell division used by most bacteria. Define a doubling time, compare the 4 phases of growth, and identify one quantitative and one qualitative method used for studying growth. Now, I will tell you, a little bit of this you will be doing in the laboratory. So later in the semester, when you do serial dilutions and count bacteria, you might want to think about coming back to this chapter and reviewing the information to boost your memory and to remind you of why you're doing that particular experiment. So the basis of population growth, most bacteria grow by process that we call binary fission, and we can kind of see it drawn out over here on the right-hand side of the panel. So basically what happens is the parent cell just starts to enlarge a little bit and makes more and more stuff, duplicates or replicates its chromosomes so that everything, there's twice as much stuff as the cell actually needs. The cell envelope will kind of pull together in the center, and that starts to form a septum, and then the cell wall will build and divide into 2 separate cells. And these are now called daughter cells. And there's some, FITC and things like that that are involved in this process. We'll learn a little bit more about that when we talk about cell division. Let's take a look at the bacterial cell cycle.
The entire sequence of events of cell elongation and binary fission, by which one bacterial cell becomes 2 cells, is referred to as the cell cycle. Because each daughter cell must receive at least 1 copy of the genetic material, DNA replication and cell division must be closely coordinated. Septum formation is triggered when the cell reaches a threshold size and DNA replication is completed. A number of proteins, including the FTSZ protein, are essential for forming the cross septum at the center of the cell and not at its ends. These proteins are scattered throughout the cell between divisions. At the onset of septum formation, FTSZ protein forms a ring at the division site. The FTSZ protein ring is a contractile structure that uses the breakdown of GTP as an energy source. As the ring contracts, synthesis of cell membrane and wall follow until cell division is complete. The FTSZ protein and other cell division proteins then disperse throughout the cell. DNA replication involves replisomes, synthesizing machinery that includes DNA polymerase, helicase, and gyrase. DNA replication consistently takes 40 minutes to complete. Therefore, when Escherichia coli is slowly grown at 37 degrees Celsius with a generation time of 60 minutes, it takes 40 minutes for the DNA molecule to be copied. This is followed by a 20-minute period during which a septum is formed, resulting in cell division. No DNA replication occurs during this time. However, E. coli can reproduce much more quickly. They do this by starting a second round of replication before the first round is complete, resulting in daughter cells inheriting DNA in which replication has already begun. Because of this, E. coli can actually complete their entire cell cycle in 20 minutes.
So I like that little video. I don't always show all of the videos that are embedded in your textbook. I think you should watch them all because I think you can probably learn from them. This one I like because it kind of ties us into replication as well, and it gives us an idea of how or why bacteria can divide so quickly, because before it finishes the division, it can already be synthesizing DNA for the next division. And this really rapid generation time can be as short as 10 to 12 minutes for some organisms. So this growth we refer to as exponential, because each time the bacteria divides, it's doubling the numbers. So you can kind of see that up here, where if you start out with one, after one replication, you now have 2 to 4 to 8 to 16 to 32 to 64 to 128 to 256 to 512 to 1,024. You can see that it does not take long to get a lot of numbers. And in fact, that's why you can go from one on your petri dish to billions in a 24-hour period, because of this really rapid ability of the bacteria to grow. And again, we refer to that as a generation time or a doubling time. Pardon me. So we can draw this in two different ways. Because it's exponential growth or growing logarithmically, we can draw it on a log table, which we see in the upper panel, and that's going to have a kind of a straight line going up. But if you look at the axes here, this is logarithmic growth. And so you can see that over the course of 20 hours, you're going from 0 to over 1 million organisms. On the other hand, if you were going to plot this arithmetically, you would kind of have sort of like a flat line, and then it'd be a steep uptick as those numbers increase so quickly. I think the logarithmic way of plotting this data actually makes it a little easier to visualize than the arithmetic. Um, so how can we calculate this growth? So to calculate the population size over time, you need to have some kinds of information. And here is the formula. So the n to the f, this is the total number of cells at some point in the growth phase. So for us, it would be like the final number, equals the n to the I, what you started with, times 2 to the n, where n is the number of, generations that you had. So let's go ahead and kind of write one of these out. So let's assume, and I'm taking this, let's assume that you had an egg salad sandwich that you left in your car, and you left it in your car for 4 hours. Let's assume that somehow your sandwich was contaminated with 10 organisms when you first made the sandwich. So your N I equals 10, because that's what you started with. And you left it for 4 hours. 4 hours times 60 minutes per hour will give you 240 minutes. So that's how long you had. Now, let's say this a generation time for the Staph aureus is 20 minutes. So if it's 20 minutes, that means basically there's three divisions, three doublings every hour. So you had 4 hours, 4 times 3 would give you 12 generations. So that's what your n is here. So you have your n to the f equals your n to the I is 10 times 2 to the n is 12. So your exponent is 12 here. And if you plug these numbers into your calculator, you will come up with 40,960 organisms after 4 hours. And if we just let it go another 20 minutes, you would have over 80,000. And in another 20 minutes after that, you'd have over 160,000. And another 20 minutes after that, you'd have over 220,000. So you'd have over 200,000 in 5 hours. So this is a really useful way to be able to count or to calculate bacteria if you have this kind of information to start with. But a lot of times we don't have that information to start with. So we don't know how much is in our samples when we start. And this is actually what you'll be doing in the laboratory. So we're going to go very quickly through this because I think that you will learn more about it when you do it in the laboratory. So you have a flask, say this is some kind of a sample, maybe you got it from some sewage water and you're trying to figure out how much bacteria is in this wastewater, or you're trying to figure out maybe even how much bacteria is in some drinking water if you have a contaminated well or something. But in any case, you want to figure out how much bacteria is present in this flask. Usually these might be foods or liquids that we're interested in. So you do a series of dilutions. We call these serial dilutions. And you add a bit of the media into this, and then you incubate for various periods of time, and then you can calculate what your CFUs are. So this is what we call a viable plate count. It's a little bit different than the standard plate count that you're actually going to do in the laboratory. But you can see by adding 0.1 mLs of sample to the tubes and then incubating them for these various periods of time, how if you look at 60 minutes, you really can't observe anything because those colonies aren't going to be visible yet. But if you go 120 minutes, you'll notice 2 colonies. We have 2 colony forming units per 0.1 mLs, and so on down the line as you increase the time and the doubling times, and more organisms then are able to grow and you're able to visualize them. So what are the stages in a normal growth curve? So typically populations of bacteria won't just grow forever. They will stop growing at some point. On the petri dish, they stop growing because they run out of nutrients or they run out of space if they're not mobile. They eat up all the nutrients that are in their media. And we can look at that when we prepare a standard growth curve. And so a standard growth curve will end up looking like this. And what happens here is you have a closed system. So you have your tube of media and you add your bacteria to it, and you don't add anything else. So you're not going to be adding or removing any other culture media to keep the bacteria growing. So it's limited by the resources that are in this tube for how much it can grow. And when we do this, we have a very predictable pattern of growth. We call this the growth curve. And it involves 4 phases: the lag phase, exponential growth phase, the exponential growth phase, and the stationary phase and the death phase. And so the way we set this up is, again, you put a small bit of bacteria into the tube, and it's a closed system. So the lag phase, if you observe the growth, you're not really going to notice very much growth in that first little bit of time. And that's because the bacteria are sort of adjusting to their new environment. They may be starting to enlarge and preparing to grow and synthesizing all of the things that they need in order to divide, but they're not yet dividing at this rapid, constant pace that we see when we move into the exponential growth phase. So in the exponential growth phase, this is where the growth is either exponential or what we call logarithmic, where those doubling times are just really rapidly growing. Um, Everybody's just kind of growing at this time, and they will continue this exponential growth until such a time that they start to run out of nutrients or waste materials start to build up, because the waste materials from cells are can be toxic. Um, Imagine human waste. Our waste materials can be kind of toxic to us too. So the waste that bacteria produce, their waste products can be a little bit toxic to them as well, and certainly not provide nutrients for them. So then they will enter into the stationary phase. And during the stationary phase, the viable cells are just trying to survive. They're they're slowing down their growth. They're not dividing as rapidly. They're kind of just trying to hang on because life isn't so good right now. They're running out of nutrients. Their density is very high, and they're starting to accumulate some dead cells. Then we enter into the death phase. And in the death phase, all of these things, these limiting factors, the nutrients get worse, the waste products get higher because you have more dead cells adding to the waste. And then you get the death phase where you start having an exponential rate of death. And usually this won't go all the way down to zero. Some cells will remain viable for some period of time, but this is where, cells are dying. So when we're studying and we want to try a new antibiotic or a new treatment, most antibiotics are going to work on some part of the metabolism of the cell, whether it's on the cell wall where the cells are building cell walls, or whether it's on some other metabolic process that the cells are carrying out. So that means that for most drug therapies and things that we look at, they're going to be most effective actually when the cells are in this active exponential growth phase, because that's when they're most likely to be affected by those particular drugs, because they'll be using those metabolic processes that are targeted. So again, I've already mentioned this, and when you use antimicrobial agents like an antibiotic, that's going to accelerate the death phase, and that's going to, of course, increase and speed up how quickly the bacteria die. And as I said before, the actively growing cells are more vulnerable. So we can also use growth patterns to correspond with stages of infection. So bacterial shedding during early and middle stages of infection are more likely to spread it to others, because those organisms are more likely to be in the exponential phase. Other ways to measure, and you'll be doing this in the laboratory as well, is looking at turbidity. And you can do this using a spectrophotometer. And a spectrophotometer is a device that you put this little tube in. We call that a cuvette. You put this cuvette in, and light will pass through, and it will measure how much light transmits through the tube. So if you have more particles in the tube, more bacteria, it'll be more cloudy or turbid. Turbid means cloudy, and that's going to block or deflect the light hitting the receiver, and so it will have a lower amount of light transmitted. and we convert that into optical density. So when all the light is passing through in a clear tube with just media and no organisms, we would have an optical density of 0 or a percent transmitted of 100. If there's lots and lots and lots of particles in here, we might have an optical density of 0.5, and we'd have our percent of light going through is 50%. So we always will do that conversion because the optical density is easier to kind of visualize than looking at the percent light transmitted. But the greater the cloudiness in the tube, and you can see here the clear, no cell growth, here lots of cell growth. You can't really even see through this tube, it's so cloudy. That means there's more and more bacteria in this tube. And you'll see that in the laboratory. So pay attention when you're in the lab and you're looking at these broths and stuff. Notice how cloudy they are and whether they're turbid or not, and whether some tubes are more turbid than others. Because if you're using those to make stains or things like that, the more turbid it is, the more organisms there are. So the more you're going to be picking up on your loops and things like that. So how do we count bacteria? Another way is we can use a cytometer. And so, pardon me, this is a special type of a slide where you have this, it's a thicker slide, and then the slide cover itself has a weight to it and a thickness to it as well, much more so than the slides that we use in our normal day-to-day class. And there's a grid that the bacteria will be on, and you can then count the cells in the grid and and count them that way. We can also use this to count blood cells. In that case, instead of just calling it a cytometer, we call it a hemocytometer if we're counting blood cells. Other methods that are automated are things like using a culture counter. So a culture counter, again, you dilute your cells in a solution, and then you put them through the culture counter. The culture counter kind of sucks them in through this like little tube, and then as they pass through the tube, there's a detector that can detect as density changes, the electric current changes as passing through, and it will count the cells that way. We can also do flow cytometry, which is similar to a culture counter, but it can do a lot. Flow cytometry can do a lot because you can use fluorescently labeled antibodies to tag cells, and then you can actually count different cells. When I was working in immunology, we used flow cytometry a lot to look at our white blood cells as well as tumor cells and things like that, much more than just looking at bacteria. So flow cytometer is a very powerful tool, well beyond just counting and enumerating bacteria. That's probably one of the least used ways that you use a flow cytometer because it does so much more than that. So some other newer ways that we can use the polymerase chain reaction, which is looking at how fast the DNA is being replicated, and you can quantify bacteria that way. This way you don't have to actually isolate or culture the bacteria. You can just put the sample into your PCR mix and add your primers that are specific to that particular organism, set it up in the PCR machine, and look to see if it's replicating and how fast you can detect it. We'll learn a lot more about PCR in Module 3 when we learn about molecular biology. And then there's new other ways to measure. So things like in food and pharmaceutical industries, they are looking to measure ATP, and measuring ATP is going to measure how much activity is happening in the cells, and that's another way that can do a rapid quantification. If you have some food sample and you see a lot of ATP in it, that's going to tell you that there's some living being in there that's generating that ATP, that's producing it. So that brings us to the end of this section. Again, you can answer these questions on your own. You should be able to. If you cannot, you should go back and see if you can find the answers either in this PowerPoint or in your textbook. And I think these these questions tend to look a little bit like your exam questions or your quiz questions. And so sometimes just the repetition of seeing them can be helpful. So that brings us to the end of chapter 9. Our next chapter will be chapter 10, and I will see you soon.